The diversity of biomolecules in the food we eat is vast, including essential molecules like fat, carbohydrates, and proteins, as well as a wide array of small molecules called secondary metabolites. These compounds are made by plants in response to stresses such as disease, climate, and predators. But there is also evidence that these molecules are good for us as humans when we consume the plant as food. The biomolecules in food come in all sorts of shapes and sizes, and this diversity means that we cannot measure all of them with the same analytical method. So within the periodic table of food, or PTFI, we've worked really hard to look at all of these small molecules in a very regimented way. This is important because while we have the technology to look at small molecules, typically the labs that are doing this all do it differently. So their answer to questions like what's in an apple, what's in a tomato, are not the same, not comparable. So this is something that in the PTFI, we've made a goal of ours is so that we can say what is similar and look at the data in more of a comparable context. Metabolomics is a type of analytical workflow that is focused on the measurement of small molecules, including secondary metabolites. The standardized PTFI method enables labs across the world to measure these molecules in the same way, so the data is easily comparable. The critical first step in this process is extraction, where small molecules are isolated from other biomolecules in food. During the extraction process, a custom internal standard mixture comprised of small molecules not naturally present in food is added to each sample. A small amount of freeze-dried food is first dissolved into an aqueous solution, which is then passed through a solid phase extraction column. This process will bind and remove interfering molecules while letting the small molecules of interest pass through. Following the isolation of small molecules, samples are dried under nitrogen to remove extra solvent and resuspended to a volume suitable for analysis with analytical instrumentation. To measure these small molecules, the PTFI uses a standardized method which aims to detect all the small molecules of food, some of which are known and some of which are not. This method involves separation of molecules by polarity using liquid chromatography and by mass using mass spectrometry. The analysis begins with injection of the small molecules extracted from our samples. The sample makes its way to a column coated with a stationary phase that interacts with the individual molecules in the sample based on their polarity, allowing us to separate the molecules in time. With so much chemical diversity in the small molecules of food, we use two methods: a reverse phase separation to resolve nonpolar molecules, and a complementary hydrophobic liquid chromatography or HILIC column to see the polar small molecules in our food. As the molecules elute from this column, they are introduced into the mass spectrometer, where a high voltage is applied, resulting in the generation of gas phase ionized molecules. These charged gas phase ions are then directed to a mass analyzer that separates analytes by their mass to charge or M/Z value prior to detection. This data acquisition can be visualized in real time by a chromatogram, which shows us the signal intensity for all of the detected molecules over time. The resulting data files are uploaded for further processing using the standardized PTFI data processing workflow, enabling evaluation of relative intensity changes for molecules that match to a global consensus library of formula and authenticated standards. With this method, our team at the PTFI is working to understand how these molecules serve the plant in defense against external stressors, such as climate change, and how these molecules translate to color, taste, and health benefits when they reach our eyes, mouth, and stomachs.